YouTube? No. Yes, it's on YouTube. So everything we say now is definitely recorded. So
you may want to start inviting people there, probably.
That's right.
Yep. I need to just mute this because it'll echo.
Yeah.
There we go.
So now we wait a couple of minutes for people to join in. All the Roelands.
Yeah. Let's see how many Roelands we can collect.
I think we should have a competition to see how many Roelands, how many Sundays do we manage to get in.
Okay, I'm just bringing people in.
Sunday, I was telling Roeland that you're contributing to the one introducing him.
Yes. Let me, what time have we got? Give a couple of minutes yet.
Yeah. I also promise that you have embarrassing things to tell about him.
It's not embarrassing, it's nice.
Oh, this is very disappointing.
I could tell some embarrassing stories, but it wouldn't be appropriate.
It's only on live on YouTube anyway.
That's right.
Embarrassing stories should become a requirement for introducing this.
Quite.
Especially if they are going to be kept on YouTube.
Okay. I think we can probably get started.
Go ahead.
Yeah. Okay, well, it's a pleasure to welcome friend and colleague Roeland Merks to give our seminar today. Of course, we'd rather have him in person, but unfortunately, that's not the way right now. But there will be future opportunities. We're promising to bring everyone back in person because you don't get the nice discussion and meeting afterwards that you would normally get if you came to visit. So Roeland has been a kind of pioneer in developing, I guess, mainly cellular Potts model-based approaches for a number of things. But I think, loosely speaking, we could sort of talk to as in relation to development. He has done specific work on cancer, and he's obviously going to talk to us about that. And one of the kind of little vignettes I wanted to give was, I remember back in Dundee, we had this library that was essentially a glorified office in a way, but it spanned 2 floors, and there was you would have proper journals, actually paper journals you could go and view. And there was this issue of non-linearity, and I know Roland's going to know what I'm talking about. And the cover of it was this amazing cellular Potts model. And I just couldn't believe that Roeland had managed to get on the cover of this journal. But not only that, he was on the cover for an entire year. It was literally every single issue. And I'm like, one day I want to be on the cover of non-linearity. So it was very impressive. And it also kind of, I guess, was emphasizing just how far sort of agent-based or cell-based approaches had come, because for a long time they definitely weren't embraced by communities like those that would publish in non-linearity, right? So it was definitely nice to see that. So let me just give you a little bit of information about Roeland. So he did his master's in biology at Utrecht University back in '93. And then he went and did a PhD in computational science at the University of Amsterdam. Who was that with, Roeland?
Sorry?
Who was your advisor for your PhD?
Peter Sloot and Jaap Kaandorp. I worked on coral growth.
Oh, yeah. So the coral, that beautiful book on coral, you were involved in that?
Well, not quite, but yeah, I knew Jaap was writing it at the time.
All right.
Yeah.
And then in 2003, you went to Indiana University, and I guess that's where you did your angiogenesis work with James Glazier for your postdoc.
Yes. Yeah.
And then you moved back to Belgium to become a young group leader in 2005. And then I guess you moved to the Centrum Visconti Informatica in 2008. And in 2014, you became full professor of mathematical biology at the Leiden University at the Mathematical Institute of Biology. So that's quite a nice vignette of his career, but I think it really doesn't do justice to some of the cool visuals that Roeland has been entertaining us with over the years. And I'm very much looking forward to hearing what he's got to tell us about in terms of cellular Potts modeling of cell-to-cell and cell ECM interactions during angiogenesis and tumor evolution. So Roeland, take it away.
Okay, thank you so much. Thank you so much for the invitation. So I'm going to talk about work by a lot of people, and you see just a sample of those over here. And I'm sorry if I forgot anybody to list, and I'll make sure the name appears on the next title slide. So what I'm going to talk about today is cancer and angiogenesis. And as you know, tumors will start to grow. Start off as small collectives of cells, and when cells become hungry in the middle. Can you see my mouse pointer, by the way?
Yes.
Okay, perfect. Yeah. So once a couple of cells become hypoxic in the middle, they start secreting all kinds of growth factors. This will stimulate cells in the blood vessels, endothelial cells. They will start migrating, breaking down the matrix, and form a small blood vessel. And then, of course, if that happens, the tumor can get access to oxygen and nutrients and can grow and also spread. And so what I started to do in my postdoc in Indiana University is to try and understand if we can understand blood vessel formation as a process of collective behavior, much like we can understand the shape of a whole swarm of birds as collective behavior. And so to do that, what you can do is also simplify the problem. And this is now what we are simplifying the problem experimentally. And this is now what we're doing in my lab. So what you see here is a video made by PhD student Professor Goese, where she takes a cell line. These are HMAC1 cells, human microvascular and endothelial cells that are labeled with fluorescent dyes. And if you put those into an environment that mimics the natural surroundings of these endothelial cells, they tend to form network-like structures, blood vessel-like structures. So what I tried to do back then in Indiana University is to see if we can understand this collective behavior by first modeling the behavior of the individual cell, and then predicting from that the collective behavior. And so that is a technique that in general is called cell-based models. Well, and people in Moffitt Cancer Center are very familiar with many of those methods, including where we put in some model of cell behavior. And this can be a very simple behavioral model or a very complicated behavioral model guided by other models, for example, ordinary differential equation models of the signaling networks. But it can also be a simple rule. And what we get out of it is a prediction of the collective behavior. This can be development of a multicellular structure like tumor or blood vessel. And what we want to achieve with that is to understand, well, first of all, how cells build tissues, but also how the resulting structure of this tissue is then feeding back on cell behavior. Because when a structure is forming, cells will get different neighbors. And this may then change, again, their behavior. But also how then changes in the rules that cells follow, for example, due to genetic mutations that may break a signaling network, how this can then lead to phenotypic changes. But also I will show you an example of tumor evolution where these mutations are happening in the same timescale as the collective behavior. So, well, there are then many ways of modeling single cells, of representing a single cell. we typically tend to use lattice-based approaches where we represent cells as points on a regular lattice, and this is just convenient for computational purposes. But since it's very important sometimes that cells have a particular shape or that they can be compressed, we typically use multiple particles for a model, and then you arrive at this Cellular Potts model that was already introduced nicely by Sandy. There are also all kinds of other methods, particularly off the lattice methods, where you just use some kind of coordinate for the cell, and you represent the change of these coordinates by differential equations or other types of equations. And of course, then you can use multiple particles for that, or boundary particles, where these are typically vertex-based models. So we typically tend to use what we call a Cellular Potts model, and what we do with that is represent one cell as a collection of connected points on the lattice. So you would have one cell over here, a yellow cell there, and a green cell over there, surrounded by some medium. And this medium can, for example, be just a cover slip or a coated cover slip. It can also be a flexible gel, and that's what I'm going to talk about later on. So for the moment, just assume that this is some kind of medium on which the cells in which the cells are living. Then cells can interact with all kinds of fields, describing the distribution of all kinds of signaling molecules, growth factors, metabolites, but also it can describe the strains in an ECM, in an extracellular matrix. And then cells can move due to balance between active forces, and these are the forces that they actively apply by, for example, contracting the actomyosin inside the skeleton, or by polymerizing actin and protruding part of the cell. Or it can be reactive. For example, cell tries to retract from an adjacent cell, and then there is a drag force or an adhesion force that would keep it from doing so. Well, and we can represent both of these forces in a Hamiltonian system. I won't go into the details right now, but you can then do things like that, like this. So this is a classical simulation of the Cellular Potts model of cell sorting, where we have green cells. These are the little blobs surrounded by the black lines. And we have red cells. Those are the cells at the outside. And they, well, based on adhesion differences, we can make them sort out. So then what can we do with this kind of model? Well, so what I'm showing you here is one of the first models that, the model that I developed. I think part of the slide is not visible, right? So maybe I should hide this thing, hide. Does everything still work? Otherwise, warn me.
It looks pretty good here, Roeland.
Sorry?
It looks pretty good, at least what I can see.
Oh, perfect. Okay. So
I have a question for you, Roeland.
Yeah.
So, and it's based on just your initial definition of the Potts. If you just want to show that the slide before again, because I think it's worth talking about this, because, you know, as someone who's dabbled a little bit in the Potts, I know that there's a key assumption one has to make, and it's to do with this concept of energy minimization.
Yeah.
Right. And so that's a critical piece for when you make a decision whether a cell moves or doesn't move or exchanges.
Okay, let me just bring up the, so is the question, sorry, I didn't get the question. Is it about the energy?
The question was just to say, it wasn't really a question, it was more to state that one of the...
Let me then just pull up this slide so I can explain maybe a little bit more about how it works in practice.
Yeah, so, because I think for the sake of those that don't know about Potts, it might be worthwhile to discuss that.
Yeah, so basically what we do is we represent this balance of forces between these reactive forces, things like adhesion, and active forces. So those are things like protrusion and contraction. And the way this works is as follows. So we have an energy term, it's a potential energy. And so very much like you could represent the potential energy of a falling object, like this glass. I'm not going to drop it on my laptop though. And so then if we want to understand how is this? Well, if we want to model the motion of this object, we can take simple differential equations, and we can describe the effect of gravity on that. And so then gravity imposes an acceleration. But you can also take a different approach, which is a Hamiltonian approach. And that is the idea that you take the gradient of the potential energy, and the gradient points downwards, because then the potential energy goes down, and you look for a way to minimize this potential energy. Well, there are different ways to do that. You can do that using deterministic equations. What we choose here is a Monte Carlo based approach. So we make little trials to this potential energy, try to move it upwards, and then it turns out we increase the energy. So then we say, well, that's not good. Or we move it downwards and then we reduce the energy. And so just by accepting all those ones that reduce the energy, then we can also describe the motion of this object. Well, that's what we do in the Cellular Potts model as follows. So we model the microscopic changes that cells have, and that's by picking a random site. And we consider a potential move into this random site. And then we look at what would happen to these reactive energies. So for example, cell adhesion, maybe this will bring the cell closer to a target volume, if you model this as an elastic object. And if indeed this energy gradient points downward, then we accept it. That means there's a force in that direction. But then there is also the fact that the cell is dissipating energy, and it's protruding pseudopods, it's contracting the actomyosin skeleton. And to model those kind of ruffling or membrane motions, we introduce random motility, and that's what we do for historical reasons using a thermal noise term, and where we sometimes accept movements that introduce energy. So that is done in this way. So sometimes we also accept those movements that act against these reactive forces, allowing the cell, for example, to pull itself away from adhesion motion and make a stronger connection with another cell. Well, if we then, so basically then we repeat this, and in practice what we do, we won't just naively choose random positions, but we will have algorithms where we can only look at the edges, making the thing much more efficient. So then what we can also do, we can integrate this, hybridize this with other models that will represent the spread of secreted growth factors, for example. And that's what we're doing here. So we assume that every red cell is a source of some kind of chemoattractant, VEGF, for example, and that the cells will then move into the direction of higher concentrations of this chemoattractant. So we have a hypothesis there that cells form these vascular networks by being attracted to one another through a chemical factor. And then we can start experimenting. For example, in this case, we have rounded cells that are attracted to one another, but and so it turns out this is not sufficient for producing networks. Then we can do the same thing, and that's just to show that the shape of cells is very important, and that's why we tend to use these cellular Potts models. Then it turns out that the exact same rule, attraction between cells, suffices for producing these networks. So at the time, what we wanted to do is to check
Roeland, sorry. How do you introduce that change then? So, because obviously your minimization is going to create circles, or that'd be the perfect.
How do I introduce the length? Basically, well, I introduce a further term to the Hamiltonian. So here I have a volume conservation term. So this says that I have an ideal kind of resting volume for this particular cell sigma, and an actual volume. And so if the cell deviates from this target volume, then I will introduce a penalty. So then there is a second, I can add a second term and say that there will also be a length. I measure into for every move, I measure the length of the cell and measure how the particular move would change the length of the cell. And then I simply have a target length. And this is a little bit more complicated than we can do in the standard Cellular Potts model, because we have to kind of have a way to instantaneously measure these lengths. And the way that we do that is by keeping track of all the raw momenta of the cell. So x squared, sum of x, sum of y squared, etc. Then from that we can derive an inertia tensor. And that allows us to instantaneously measure the length. And then we also need a conductivity constraint. Because otherwise the thing will simply optimize the inertia tensor and it creates two small blobs. So it's basically the short answer is we use the length constraint.
Okay, that's what I thought. Thanks.
Okay, so what we did at the time is to try and see whether we can whether these rules suffice to match the in vitro cultures. And what you see here is a culture of human umbilical vein endothelial cells, UVECs, in matrigel. And what you see is they quickly form these networks that slowly coarsen over time. Well, at the time we also tried to videotape those, but and indeed I have VHS video of these vascular networks, but the quality was too low and the computing power was also not sufficient to try and analyze these automatically. So what we did instead is just take single pictures and measure morphometric properties such as the number of branching points and so forth, but also other measures to that we could compare with the in silico models. And it turns out to match pretty well. So before showing what we can do right now, I want to go a little bit into how we can use cellular Potts modeling to understand tumor growth, and in particular tumor evolution. And I want to keep this in the context of vascularized tumors. So what I'm modeling now is to sorry, I just have to try and pull up one of the slides that I missed. Where is it? Well, anyway, it doesn't matter. I have a nice vascular normalization slide that somehow didn't make it to this presentation. I'm sorry, but anyway. So basically what I'm interested in is how will tumor respond to the vicinity of vasculature that will bring oxygen and glucose to it. So what we're doing now is to, and then I'm interested in seeing whether using therapy to normalize the vasculature so that the blood flow will be nice and steady, as opposed to a tumor type vasculature where it's very noisy and very irregular and tortuous, whether how the tumor evolution process will, or tumor plasticity process will respond to that. So what I have here is a very minimal model of a tumor. I model only the cross section. I assume there is some vasculature running through it, and I model those as point sources over here. So these are the red cells in the middle, 4 red cells. And I also assume that tumor cells are embedded within a healthy tissue. So then in every cell, what I do is that I assume there is some glucose and oxygen uptake. And we also have fields of glucose and oxygen. That if the cell becomes hypoxic, because it has too little, when it has too little oxygen, then it will start expressing HIF1 alpha, which is hypoxia-induced factor, and which will shift the balance from respiration, from using glucose and oxygen to, basically using oxphos, to fermentation, which is a much more wasteful way of using glucose to produce energy. And then when it's using respiration, then as a waste product, it will also produce reactive oxygen species. These reactive oxygen species will stabilize HIF-1 alpha. So meaning that if a cell is in a very, environment high in oxygen, a physoxic environment, then it will typically start, kind of relying on respiration. But also we assume that the ROS, so the amount of energy then the cell produces will induce growth, but we also assume that ROS will inhibit the growth. Well, so I have a couple of equations of how we do this. I can briefly go over this, but for the details, I would refer you to the paper because I have so much more to tell. But basically, what we do, what we assume is that we have a metabolism, we have a very simple model of metabolism consisting of respiration and glycolysis, which is a wasteful way of doing metabolism. Then we have a function that says, well, so much energy has been produced in a single time step. That then depends on how much respiration there was and how much glycolysis there was, where respiration will produce on average 38 ATP units of energy per molecule of glucose, whereas respiration will only produce 2. Well, then, of course, the whole thing depends on how much the cell has been able to take up. And this, again, depends on the amount of glucose available, but also on what we call an intracellular growth signal. In reality, I'm trying to simplify the whole model a little bit. In reality, this is the amount of GLUT importers expressed on the cell, and on another couple of other parameters, including the current volume of the cell. Then we also have the glucose uptake, and this will depend again on a parameter that says, what's the expression of HIF-1 alpha? In other words, how much respiration does the cell want to do versus how much glycolysis does it want to do? And of course, if it relies only on glycolysis, then it will not take up oxygen, otherwise it will take up oxygen. So then we have a function that says, okay, how does the growth then depend on the metabolism? And basically what I want you to take from this is that the amount of growth depends on how much energy was produced. It also depends on whether the growth is kind of inhibited by pressure from surrounding cells. And also it will depend on how much damage is done by the reactive oxygen species. Well, then we have this internal state. This is the HIF-1 alpha expression. This depends on current state, but also on a little bit of history. Well, then if we put all of that together, what we have is cells that are trying to grow, and they need oxygen and glucose for that, but they can also switch to using pure glycolysis. And if in the most healthy state, what I have now is that the cells form homeostatic tissue. So they grow around blood vessels. You see some cells dividing. And we have simple cell motion. We have growth and division. Pressure inhibits growth. We have cell rearrangements and some spontaneous cell death. And the endothelial cells, we basically, these are just fixed blood vessels. We don't have any morphogenesis of blood vessels in this model at the moment. And they are assumed to be sources of glucose and O2.
Roeland, can I just ask, I don't know if you're aware of the work that Mark Robertson-Tessie in my lab did, that he published in Cancer Research, and he has a metabolic model, but this is using a CA rather than cellular pots. But one of the things we were looking at there was acid-mediated invasion. So I see you have lactate in your model, but do you track that?
Yeah, I track it. Yeah.
Does it modify anything in your model?
Well, we don't do any breakdown of matrix in the model, but it modifies the I'll come back to it later. It doesn't,
Okay.
at least it doesn't break down any matrix or so.
Right. Well, it's not really the breakdown matrix, but the acidity normally hurts the normal epithelium, right? Like normal epithelial cells don't like high acidity.
Right, exactly.
So, but one other question there was, you didn't really explain it, but I know you're trying to, you get through a lot, but I'm intrigued because obviously you've got multiple diffusibles here, and then you've got your Potts sort of lattice. So are the individual components of each cell equally producing or consuming certain amounts of nutrient and producing that lactate? How do you integrate across the cell? What do you do?
Okay, so each cell's the uptake of glucose and the uptake of oxygen depends on the amount of glucose available in the surroundings, and it also depends on the amount of oxygen available in the surroundings.
Sure.
Well, further away from the blood vessel, they will take up less glucose and less oxygen, simply because it's not there.
Sure, but my question was more, how do you connect the scale difference, right? You've got your diffusible, which is on presumably a finer lattice, and then you've got your Potts cell, which is made up of individual lattice sites. So do the individual lattice sites each individually consume or produce?
Oh, that's a good question. I don't remember. I'm sorry. So your question is, do you integrate? Do you have? Maybe I can see it from. I honestly don't remember at the moment.
And look at the paper. It doesn't matter. I just thought. Yeah. Just intrigued.
I'd have to double check. I suppose that in this way. So your question is basically, if you have a bigger cell, does it consume more oxygen? And I think actually it's regulated per. Let me see. I think we model per letter side, but I really have to double check to be absolutely sure.
Okay, no worries.
It's been a while.
Yeah, it was 2017.
Yeah. So yeah, what we can keep track of is all kinds of things, including glucose, lactate, but we have no lactate at the moment because there's only respiration in this model, in this state, in the homeostatic state, and but we don't keep track of other metabolites in this model. So then we can model tortuous vessels, and we can say, well, the way we model that is by assuming that tortuous vessels will show up as a fluctuating source. So we do that by having a vessel blocking probability every time step and a vessel deblocking probability, and this then leads to a fluctuating source. And just for clarity, just to keep a continuum between blocking and non-blocking, to have a completely open vessel, we model that as a super high blocking probability, because then on average the vessel will always be open. Otherwise we get the discontinuity near probability 0. So, but basically what you see now is that in completely healthy environments, genetically stable environments, these cells will simply stay homeostatic, these tissues. And the amount of metabolites starts fluctuating, but it doesn't really affect the cells. So you get fluctuation of, you get some lactate production if there's a lack of oxygen, but when the oxygen is taken away, the lactate is taken up, and it goes back to 0. Let me see. Yeah. So, of course, what we wonder is what's going to happen if we now let these cells become genetically unstable. So what we did here is really make a completely explorative model. So we didn't want to put in any hypothesis a priori. So we took all the possible parameters we could find in the model, and that we had introduced in the model, and let those mutate. And so you have a couple of those over here. You have the incompressibility, division volume, the cell adhesion. Whoopsie, glucose. We had chemotaxis towards glucose, towards oxygen, towards lactate, and so forth and so forth. And then what you see, and unfortunately, I wish I had more, I'd say more fine-grained and slow movies. But what you see is that the tumor cells die out really quickly. And what I hope to do now in the next few slides is really explain you what is going to happen. And so basically what you find is fermentation even in the presence of oxygen. So you get what we call the Warburg effect, evolve automatically from the system. Whereas if we have normalized vessels, then I'd say that the tumor cells don't die out, and normalized vessels, meaning a higher blocking probability leading to stable source, which will lead to highly glycolytic cells that are kind of pushing out the respirating cells. And so what's happening over here? So what I'm showing you now is so is the fact that the cells try to, of course, they have to compete for growth, and of course the higher growing cells are always kind of pushing away the lower growing cells. So you get a competition for growth and also competition for being as close to the vessel as possible. So what you then see is that you quickly see the number of cells going up. Then what you see is that the number of cells goes down because they're kind of pushing away they're kind of hijacking blood vessels and taking up all the glucose. They are all going to use glycolysis and inhibiting the access for other cells. But then, and so basically what you see is a quick evolution towards glycolysis. So you see an emergence of the Warburg effect. So fermentation even in the presence of oxygen. So now, why then do the tumor cells with tortuous vessels disappear in this model? Well, that's simply because if this evolutionary process is going on where all cells really needs to have a steady supply of lots of glucose, then if you're blocking suddenly the amount of glucose, this will lead to necrosis. So even if this system rapidly evolves towards the Warburg effect, they're really sensitive to these tortuous vessels. Well, then what I always like, so this is what you see. You see lots of lactate being produced. You see, the number of cells is fluctuating highly. The oxygen is first dropping, but then it's slowly coming back because just the number of these cells is going extinct. And not because they're using aerobic glycolysis. So what I always like is to kind of debunk the story once it's built up. And indeed, what we next tried is to see, well, suppose that what we know is that a lot of this tortuosity in these blood vessels comes up from blocking, from pressure in the tumor. So if we now assume that if the cell, the blocking probability depends on the local density of cells, so that the vessel will be blocked if the density of cells is very high, but it will be released again when the pressure is released, then we see that the whole story breaks down. So what I want to show with this is how we can use a very exploratory model where we let all kinds of different parameters evolve, and how then we can start setting up hypotheses about how competition for glucose can lead to a Warburg effect and can stimulate the evolution of how to say cells using glycolysis. But then again, how a feedback from the higher level of the tumor itself can kind of turn back this effect. And of course, here I kind of show the completely other extreme where there's a full dependency of tortuosity on the blood vessel release. So I'm skipping a lot of detail of this model. And the reason is that I also wanted to show you some work that we did on the evolution of collective cell behavior. And why am I showing this in the Math Onco seminar? Because I think we can also see this as a question of how collective metastasis can emerge in cancer. So we've done this work primarily to ask how multicellular organisms evolved. But once you have cells that kind of start going their own way, then is it possible that these cells start again collaborating? And this need we see a lot during metastasis, where you see cells migrating together, for example, along blood vessels. And so can we now understand using evolutionary models how collective migration emerges? And so what we did is work by Enrico Colizzi and Renske Vromans. What we did here, what they did here is to set up a kind of arena that for them was a noisy gradient towards a food source, but you can also see it as a noisy gradient towards a blood vessel, where cells have to migrate towards the food source. And the cells then closer to this hotspot, read maybe blood vessel, they reproduce most efficiently. So what they did, they had receptor ligand pairs, where so each cell had a kind of receptor and a ligand, and the match between this receptor and ligands gives an adhesion strength between the cells. Then also they have a model for chemotactic in noisy gradient, where cells by themselves, they always have a particular movement direction, and maybe I have to switch on the light. It's getting very dark here. So, so cells on their own, they can feel a very noisy gradient, but not very efficiently, and they behave as persistent random walkers. So then, indeed, in the initial situation, chemotaxis is very inefficient. So the cells are migrating; they behave a little bit like metastatic cells, like mesenchymal cells, and they migrate through this gradient. But
Roland, how is the chemotactic direction decided by a cell, given that it's got, like, the center of mass or something?
Yeah, so the way they do it is by setting a persistence direction. That's this theta, and the cell tends to move towards the theta. And they do this in the Cellular Potts model by favoring copy directions along this theta direction and reducing copy directions against the theta. So then the theta directions updated by gradients, the cell feels. But since just by the way the gradient is laid out by particles, the reorientation of theta is very imprecise.
Okay. So basically the center is changing depending on the geometry of the cell, and it looks all around it.
So it finds the center. It finds the center of mass of the mass distribution under the cell, right. And that determines theta.
Okay, thanks.
Okay, so what you get then is that cells, but you could do this in many different ways, right? And so then what you get is kind of rapidly migrating cells. Well, now the amazing thing is, and this is also known from previous work, for example, work by Brian Campbell, is that if you push a lot of these cells together, you let them adhere, then even though the chemotaxis of individual cells is very noisy, collectively they start migrating. They are really good at chemotactic. And so what Renske and what Enrico and Renske next ask is, now what would happen if you let this evolve? Oh, so this is just a little bit of than the single cells. And now, yeah, okay. So then they asked, can this collective behavior evolve? And they did this in the following way. So they said, okay, let us have seasons, but you can also see this as a tortuous tumor where sometimes blood vessels are blocked, and a tumor with tortuous vasculature, where sometimes one of the vessels is blocked and then another opens. So they modeled this as seasons where the gradient changes orientation. And then at the end of the season, they killed half the cells that you can see in figure B, and they let the cells reproduce dependent on the distance from the gradient origin. So this is the reproduction stage, and now it has to move to a different direction. And along the way, they also mutate the ligands and receptors such that the adhesion is changing. Now let's look at what happens. So you started with a very noisy, a very radically radically migrating population of cells. But as the gradient is moved, or as the location of the open vasculature is changing, slowly they acquire the ability to migrate really efficiently by working together.
Roland, is that little kind of line in the cell telling you the cell's orientation?
Say it again?
Is the little line inside the cell telling you the cell's orientation?
Exactly. Yes. So that's this vector theta. So what you now see is that indeed these cells can acquire the ability to adhere to one another and hence migrate more efficiently. But it doesn't happen in all circumstances. It turns out that this depends on the length of the season. So now if you have a very short season, then it's just efficient to just migrate wherever you can and just migrate really quickly, so that maybe you're lucky and you're close to the source and you get to reproduce. But then if the seasons are very long, there's plenty of time to migrate, and then it's better to migrate collectively, because the collective migrators will always win over the single migrators. But then if there's also this interesting point in the middle, and the gamma is a kind of aggregate parameter to show the aggregation, where a high value of gamma indicates aggregation and a low value indicates dispersion. And you see there is actually a bistable region where they can either move to unicellular or they can move to multicellular behavior. And so what happens? Well, they figured out that this has to do with interference competition. Now, suppose I have here the peak of my gradient. And I
Roland, I had a question.
Yeah.
We go back one slide.
Sure.
Where do you start the population, at 0?
I start the population. I initiate the population at the negative gamma.
So they're unicellular to begin.
Well, then no, they're a single cell to begin with.
No, but I mean that on this green, for example, they start unicellular, and so if it's a short season, they stay unicellular. Would you say?
Exactly.
Right. Have you tried the opposite where you start them with the multicellular components with a short season? Do they lose multicellularity, or is it simply there's no pressure to become multicellular when the season is short, but it would be okay?
That's an interesting question. Let me think about it. I don't think we've tried it just exactly like that, but I'm wondering if we can derive the answer from another. Let me just continue. Maybe I can answer it from the following. But I don't think we've answered it like just exactly in this way. No, it's an interesting experiment. Yeah. So so basically what they did here is they started with a single cell migrate, unicellulars, and a region of single cells of multicellulars behind, the blue ones. And then, even though these blue ones would be able to migrate really quickly to the peak of the gradient, the single cells are blocking them. Whereas if you do this the other way around, then they can still easily migrate. So so what you see here now is let me see here. So now they compare adhesive cells, and they make collectives of these non-adhesive cells and the adhesive cells, and either put the adhesive cells in front and non-adhesive cells in the back, like this. And then, of course, the adhesive cells can quickly run away. But if you put them the other way around, then they can't compete. And so, indeed, this explains this bistable region, because it really depends where now the non-adhesive cells are. And indeed, yeah, interesting question. I don't think we can derive it from these experiments. Good question. I will ask them. So, are there any questions so far about these evolutionary studies? If not, then I would like to go a bit more into...
I have one, Roland, sorry. I guess I'm intrigued about exactly when you say that the receptors evolve or something like that. I mean, what exactly, what's changing on the cell during evolution?
Yeah, so the question basically is, what is changing on the cells during evolution? Is that right?
Correct.
So basically what they implemented is that every cell expresses ligands and receptors. And so basically the match between two cells, the adhesion between two cells, depends on the amount of receptors and ligands that are matching in this way. Right, so if they are exactly complementary, the receptor and ligands are exactly complementary, then they match really well, and the adhesion is very high. So we set a relatively low J value. But if none of them match, then they are single-celled. And so what is mutating is the presence and absence of these receptors. Does that clarify?
That really helps. But of course, cells interface along multiple subcellular regions in your pot cell, right? So is it the entire cell that has that same receptor ligand, or is it just the local protrusion?
No, it's really the so we assume that that one cell, one particular cell has one particular set of receptors and ligands. So we don't distinguish between different regions of the cells.
And so that is represented by this binary string here. And is that what it is? Is it 6 characters long, or is it, how did you choose that? What size did you choose?
They didn't really vary it.
Because obviously you can make it easier or harder to be fully adhesive for,
that's
right?
true. I think we didn't do a full study of the length, but it's I forget the exact length. It's longer than what is shown in this picture.
All right, okay.
That exact length. I would have to look it up in the paper. I'm sorry.
No worries. Thanks.
All right, but I agree with you that this can change the rate by which it evolves and how easily it evolves. That's right. Yeah. So what we've ignored in all these models is a very important component of tumors and tissues, and that's the extracellular matrix. So, well, I think most of you know what the extracellular matrix is, but it's basically a network of materials that cells produce for mechanical support, but it can also be a medium for signaling. Well, so first of all, the presence of or absence of ECM can act as a signal, but it can also be chemical signals can bind to the matrix, but also pulling forces and strains and stresses in the matrix can really act as signals. And these signals can be, I sometimes call them written signals, because they can be really long-lived. The cell can drop chemicals and walk away, and then another cell can pick them up. But it can also be long distance, because a cell can pull collagen fiber, for example, and a couple of cell diameters further on can pick it up, or can even generate strained fields in the whole tissue. And so that's really why we shouldn't ignore the ECM in our cell-based models. And so what I'm going to show you now is first some single cell approaches where we try to model the ECM in more detail. And then I'll switch to some collective cell behavior, but it depends a little bit on the time I have, because it's already almost 1 hour. But you have to stop me, and otherwise I can just show you some pretty pictures and some references. So basically, what we first started doing is exploring a little bit of how the ECM can be a chemical signal. So how it can help coordinate cell behavior during angiogenesis, and this is in fact very similar to the model that Sandy Anderson developed together with Mark Chaplin. But here, what we do is we try to do this from a completely cell-based perspective. So what we have here is a couple of components that we add. And well, to cut a long story short, what we assume here is that cells secrete MMPs. They have an optimal concentration of ECM where they can migrate. They migrate towards a source of VEGF, and they also proliferate under tension. So we worked a bit more on this to take this from a more explorative model to a more model that closely matches a widely used microvascular endothelial vascular assay, where endothelial cells are migrating into a fibrin layer. And basically what we did there is study the effect of all kinds of additions to interfere with the plasmin system that will break down fibrin. But what I wanted to spend most time on is to discuss how the mechanics of the extracellular matrix, how we should model the mechanics of the extracellular matrix. And indeed, it's known that the stiffness of the extracellular matrix affects vascular patterning. And this is an example of this. These are experiments from Cynthia Ranitz-King's group, where she's culturing bovine aortic endothelial cells on top of a matrix, synthetic matrix, of which you can model of which you can control the stiffness really well. And what you then see is that on very soft matrices, these cells still form structures that look like these nice vascular networks that we've seen, but on very stiff matrices, they form monolayers. But you'll have to stop me when time is up, right? But I can either show you some older work, or I can.
You're fine. Continue, Roland.
Okay, all right, okay. So basically what we start with is to try and explain some single cell behavior. So where we try to explain the following observations, where the shape depends on substrate stiffness. So many cell types, including those cardiomyocytes, they have the tendency to become really small and round if they're cultured on top of soft matrices. But they tend to spread out like pancakes if they're cultured on very stiff matrices. And on intermediate matrices, they tend to elongate. And so we asked ourselves how this can work. And what we did, well, one of the structures that mediate those interactions are the so-called focal adhesions. And focal adhesions are structures that link the actin cytoskeleton to the via integrins to the extracellular matrix. And the interesting properties of those focal adhesions are that they strengthen under force. So the more force the cell applies to these focal adhesions, the bigger and stronger they become. But if the force is released, they slowly are broken down and they are released. And in this way, they act really as a cluster of so-called catch slip-ons. So that means that if you are applying a lot of force, they just break, of course. But if you apply a little bit of force, they become stronger. And you can see this well, I always try to understand this in this way. I'm not sure if you can see my hands, but suppose you would have two little hooks and you are in a very noisy environment, and you can imagine that under a little bit of force, you can easily slip. But if you start applying force, then the noise will have less of an effect on that. And then if you increase the force again, then of course you will slip again. So what we did to model this is to take again the Cellular Potts model and then to model, first of all, the traction forces that the cell will apply on the substrate. And we used for that a published empirical model, which is called the first moment of area model, that was introduced by Lemon and Roemer, where, and basically the idea there is that the force on a particular part of the cell is proportional to the distance to the center of mass. So the further away, if the cell is a long protrusion, then it will apply a lot of force over there. So these forces then we feed to our focal adhesion models that are growing under force. And if the focal adhesions are bigger, the probability that they will retract themselves from the substrate is going to be higher. And so in this way, we can kind of switch between CPM, the traction forces, and the growth of the focal adhesions. And these are all modeled using ordinary differential equations. So the result is then that on stiff substrates, the FAs grow stronger and they're ripped from the substrate less easily. Oh. And this is then the result. So what you see is an animation of these movies. The cell at random becomes bigger. If then some parts of the cell are a bit further from the center of mass, then it manages to kind of get a hold onto the substrate because the force is large and the focal adhesion grows. So that allows the cell to expand a little bit further, and again at random generates some extra protrusions that will again stick to the substrate. And so this generates a kind of ratchet effect by which a cell can go spread out on stiff substrates. And so indeed, then on soft substrates, you see that the cells become relatively small and they form hardly any focal adhesions. But on stiff substrates, you see that you get a ring of focal adhesions that matches also some experimental observations, which are kind of gluing the cell to the substrate. So now I've explained the very small cells and I've explained the very big cells, but I haven't explained why the cells become elongated for intermediate stiffnesses. And so for that, it turns out that we need to add a further complication to the model. We also need to introduce the deformation of the ECM. And so what we're doing then is to, again, we have our Cellular Potts model. We again have the force model, the first moment of the area model. And then we also have a finite element model to generate, to model the strains in the ECM. And so basically what we can do there is we calculate the forces that the cells are applying. We feed those to a finite element model. We let it run for a while. This generates strains and stresses that are then going to affect again the focal adhesions. And they do this in the following way, that if the substrate is strained, then this reinforces the focal adhesions, and this is due to recruitment of stabilizing proteins. And this is according to a previous model that is there in the literature. So basically what we do is kind of combine models of different parts of the cells, different scales of the cells, and try to see if we can predict the behavior of the whole cell from that. And indeed, then what we get is this. So the colors show the amount of the strain energy density in the substrate. So if the substrate is more red, then it is more strained, and that there the focal adhesions are reinforced more. And what you now get is a kind of cooperative effect where the strain generated by one focal adhesion will directly also strengthen adjacent focal adhesions, because they also respond to the planar stress. And then over time, you can see that an instability occurs and the cells start elongating. Well, then we can, of course, vary all kinds of parameters. For example, we can vary the speed of ectomizing contraction and change the stiffness at which these cells are elongating. And what I like a lot about this model is that we get for free a couple of other behaviors of cells. And one of those is that cells tend to move towards higher stiffness, a phenomenon called durotaxis. Somehow the movie never wants to play completely. But basically what is happening here is that the cell slowly drifts towards the more stiff part of this gradient. We have a gradient from very soft to very stiff. And the idea is that, the mechanism is that if focal adhesions are continuously breaking and forming, and they have a higher probability of becoming bigger at the stiffer side of the cell than at the softer side of the cell, then the focal adhesions on the stiffer side are more long-lived. And as a result, the cell will drift towards higher gradients. And indeed, matching experimental observations, what we see is that the cells move to higher stiffness, but also the cell speed is going to depend on the slope of this stiffness gradient. So then, of course, you may wonder, well, I put in some empirical model, and I will kind of rush you over these slides because I want to show you some recent developments that we did on modeling the extracellular matrix. But basically, you can, of course, wonder if the model of intracellular contractility is correct. And one of the problems there is that we assume the cells are internally isotropic. So this is why we teamed up with physicists in our department, Luca Giomi in particular, and we worked both with Koen Schakeraad, a PhD student, and who was modeling the effect of anisotropic contraction inside the cell. So basically what he did is build theoretical models of how contraction of the cytoskeleton is going to affect the boundaries of the cells. And well, one of the classical models tells you that the cells will form circular arcs, and that these circular arcs will depend on the distance between two adhesion points and on the conductivity strength. And indeed, this is called the Young-Laplace law, and we can model this in the ECM. We work with a slightly different formulation where we don't have a target area anymore, but we just say that the cell has to contract to zero. This gives isotropic contraction of the cell. And then indeed, we can nicely predict the Young-Laplace law from the Cellular Potts model. So what we then next did is to model the anisotropic contraction. And to do that, what we integrated inside the Cellular Potts model is a model called the Q-tensor model that gives a description of, first of all, how abstract particles, but these could be actin filaments, how they are either aligning or completely isotopically distributed. And at the same time, it also gives the orientation. So it gives both the order parameter of the fibers, and it gives also the orientation of the fibers. Well, then if we turn the cellular forces and translate this in a Hamiltonian, which we can do because the two are entirely interchangeable, then we can change this into an active motility term that we call also sometimes a work term in the Cellular Potts model, and then predict also isotropic cells, anisotropic contraction, and indeed these predictions match really well the theory that Koen did on this. And now the nice thing is that whereas the previous simulation and the vertex-based simulation that he did, they don't allow for very complicated cell shapes. But with this model, we can now model more complicated cell shapes. And for example, these cell shapes on patterned substrates like this, how do you call this shape? The bent T shape. And indeed, this new model now leads to some improvements of the predictions of the cell forces, in particular at this part. And this is due to the alignment of fibers in the middle. And these are two parameters that change the amount of alignment of the fibers to one another, and also the amount of alignment to the boundaries of the cells. Well, so then we can also do this dynamically. So if you can give me just a little bit more time. By the way, I like it how this one is now aligning along to the boundary, and then you see this last contraction of the cell boundary. So because so far I modeled the extracellular matrix as a isotropic material, but in fact we know, and I showed this with this first example movie, that ECMs are very fibrous. So can we now introduce another way to also model fibrous extracellular matrices? And this is a thing that we're now working on, and I hope to have a preprint of this out very soon, where we are using now molecular dynamics codes to model the extracellular matrix and integrate those with the Cellular Potts model. Well, now you might ask, why not use something like a molecular, subcellular element model, so you can do everything in the molecular dynamics framework? Well, the reason is that we have now developed all these tools that we want to combine with one another. For example, to have isotropic and anisotropic contraction together with fibrous ECMs, together with directed migration, for example. So what I show you now here is if my movies want to work, I have a single contracting cell that is forming a nice and round cell. Now I'm reducing the amount of cross-linking. We have fibers that are cross-linked to one another with very stiff springs, and this is a thing you can chemically quite easily change. And so what we find now, that if we have a cell embedded in a high cross-linking gel, that of course the gel is very resistant, and the cell, if the cell tries to contract to zero, the gel nicely keeps it back. But now we try to do the same thing in a low cross-linking gel. The cell tries to contract, there are only a few bundles that hold the cell back, and you get again also these nice arcs that we see in these different other models. So now we can also take these simulations and time average the density of the fibers at every lattice site. And indeed, what you then see is, well, first of all, pictures that look very much like the microscopic pictures, but you also see densification around the cells, which is observed in the low cross-linking gels. And you also see that you get bundles. And to further study how these bundles will then affect the cell density, now what experimental biophysicists will do is to do atomic force microscopy. I'm a bit ahead of my story. There you go. is use atomic force microscopy, where you can kind of take a cantilever and push into the gel and check how much is the deflection. And what you then get is indeed, if a cell is pulling on a substrate, then you get a long region of very dense and stiff matrices. And you see a particular distance of which the matrix is strain stiffening. So what we did, and we is Erika Tsingels and Bente Hilde Bakker, together with Herman Jan Hupkes of the Mathematical Institute in Leiden, is that they also did an acetical atomic force microscopy, where they tried to push an MD particle in this strained gel. And indeed, what you now see is that along these places where the fibers are highly strained and where the noise is very low, where they form bundles, then indeed the stiffness is going to be much higher than elsewhere. And also, if you now quantify this, you see that we can recapitulate quite well this decay of the strain stiffening away from the cell. Well, now, of course, we are interested in upscaling this to multicellular systems. So here you see two examples of contracting cells. And already you see this kind of bundle formation that is observed experimentally. We see it again in these ab initio simulations. Well, I'm kind of well over my time, but I will just show you some final slides on how we can upscale this to the collective cell behavior. Here we use, again, the isotropic model, and to show that these interactions between cells and extracellular matrix are indeed sufficient to produce collective cell behavior, both formation of vascular networks and sprouting. Well, then, now I have a couple of models. Which one now is correct? Well, the jury is currently out, and there are many more models, some of ourselves and many of colleagues. And what we're now doing is we set up an experimental lab where we do cell cultures and do timeless microscopy together with image analysis to start analyzing in more detail the development of how the number of branching points and all the morphometrics changes as a vascular network is developing. And we can do this now much better than I could during my postdoc, where I had to kind of take these dishes in the middle of the night out of the stove and make pictures and then do some image analysis on single images. Now we can do this for whole tracks, for whole time-lapse images. Well, thanks to Tessa Verhoeven and Babette, Babette de Jong, and also the work of student Vincent Vermeulen, who wrote the ImageJ scripts. And so now we can just do this automatically, and we can of course also do this for our simulations. And I hope to tell you pretty soon which of my models I should reject, which models I can still save with a little bit of adjustments of parameters. So with that, I would like to stop, and I'm ready to take any questions.
Thanks, Roland. That was quite a tour de force of cellular pots from bridging many scales. So I asked many questions, but I'm going to give the chance for others to ask if they have some.
I'm happy to kick us off on a philosophical side. Hey, Roland, that was absolutely beautiful. Thank you so much. And so from a philosophical end, I mean, you've shown quite beautifully that some of these simulations can qualitatively and visually mimic some of the experimental results and data that you have. Would it be possible for you to maybe comment on how easy or not easy it is to calibrate and validate these kind of modeling approaches?
Yeah, thank you. That's a very good question. The answer is of course not very precise because it's and the answer is that sometimes it's possible and sometimes it's not possible. In many cases, it will not be possible. In some cases, for example, these how do you say these ones. Yeah, then we know the properties of the gel pretty well, so we can calibrate the molecular dynamics models pretty well. We can look at shears, and we can look at cross-linker densities, and we're able to quantify the forces that cells apply, and we can quantify the response of the gel. So in those cases, it should be possible to calibrate these quite well. But of course, if you are working with whole cells that are behaving collectively, and you're looking for responses to changes in the gels, then things become really difficult, right? So then it's becoming really hard to validate a model really based on quantitative agreements. And that's when we are looking for qualitative agreements. And so that's basically what this slide is explaining. So then we would look for things like, okay, how are we looking now for, how do you say, does the shape of the curve, if we look at the number of branching points, for example, in a developing network. So does the number of branching points, does it change exponentially, or maybe linearly, or maybe quadratically? And do we have then models that we can distinguish on having different shapes of curves? So then those are qualitative differences between models that we can test against our in vitro models. And another thing I'm really interested in is to see if we can if we can really focus on qualitative responses of models. And this is what I mean by this slide. So we can now have our experimental models and, for example, change the stiffness of the gels, or change the density, change cross-linking density, all these kind of things. And we see that the curve of our time-lapse images responds in a particular way. Then we can compare those responses to our models and fine-tune the models or reject the models based on the difference between those two. And this is indeed what we're now working on, is to really go back and forth between our experimental model, then go back to the mathematical model and see if we can, we can really pinpoint key differences where the models are really wrong, and then suggest changes to plausible changes, or plausible new rules or extra rules. Think of cell differentiation. Think of quantitatively, maybe several cell types, cell differentiation, things like MMP secretion, all these kind of things that can modify the behavior of cell culture.
Yeah, excellent. Thank you so much.
So just following on this philosophical vein, this was very impressive, and clearly you've been working on many different sort of studies on the use of these AM-based models. Is there an ultimate goal for some of these, or is just and I don't think there's a problem with that. I think basic science is absolutely great, but is this going in a direction that has an application for biologists or clinicians?
So your question is there an application? So I think the application in this work is twofold. So on the one hand, it's really basic science. It's actually threefold.
That's right, yeah.
On the one hand, it's basic science. I just want to figure out how cells build blood vessels. And if I understand that, I hope that I can also say something more about different systems. I can maybe say something general. Then, of course, the second thing is we hope to find eventually, but this is really down the road, is we hope to see the ECM as a target for therapies. So really by understanding how the physical environment of a tumor cell really is affecting also morphogenesis and the ability of cells to form blood vessels. Maybe we can use the ECM as a target for normalizing blood vessels. So that's of course, that's certainly a more long-term goal, but of course this idea is all kind of woven through this type of work. And then there is another thing, and I think that is really goes back to this philosophical question, is can we now also develop new methodologies of how to integrate mathematical models in these type of research questions? And so how can we use our models to that we are sure are super wrong, and they're much more wrong than they are in different fields, because we have no clue what's really happening. There are always many more components that we didn't include than there are components that we did include. So we are always terribly off, and still can we use these models to get some insights, non-intuitive insights into the actual system that will bring us new ideas. And I think that's finding a way of finding a systematic way of doing that. That's also one of my goals.
Very good. Thanks, Roeland.
Okay, well, if there's no more questions, then thanks, Roeland, for an entertaining trip through the world of cellular Potts modeling. And hopefully we get to see you again in person in the not too distant future.
So, yeah, would be great. Same. See you soon. Yeah. Okay.